Okay, I think we've got everybody, and good afternoon. I'm really pleased to welcome you on this very special occasion for our School of Medicine. It is always deeply meaningful to have events that celebrate important members of our faculty and our alumni whose generosity makes it possible to establish endowed professorships. I think it's because we are so busy and focused on the next set of accomplishments, the next set of results, the next publication, the next grant. And so these events allow us to think back and treasure what has been done already. And it could not be more timely for us to memorialize what we have done at WashU Medicine to advance basic science as a global leader. Today we celebrate the history of our Department of Biochemistry and Molecular Biophysics and the creation of the Carl Frieden Distinguished Professorship. We honor the legacy of Carl Frieden, a remarkable scientist, mentor, and colleague, and celebrate the generosity of an alumna whose career has been shaped by his influence. I want to extend a warm welcome to Dr. Nan Ziyang. Nan, we are so delighted to have you back at Washington University, and we also welcome Jean Lei, Nan's colleague and co-founder of BioLegend and president and CEO of Legend Foundation. And also, I want to just mention, we have Chancellor Martin here with us who will join me shortly in bestowing medallions to Dr. Ziyang, Dr. Lei, and Dr. Frieden. Let me say a few words about Nan. She earned her master's and PhD at WashU. While pursuing her PhD, she worked with Dr. John Stephen Taylor, a professor of chemistry, in an interdisciplinary program that blended chemistry and biology, something that deeply resonated with her passion for using chemistry to solve real-world human health challenges. Following her PhD, Nan joined Dr. Frieden's lab at the School of Medicine as a postdoctoral fellow, truly embodying the connection between WashU's two campuses and the spirit of collaboration that we so value. Today, Nan is an enormously successful leader in the biotech space. She has helped drive innovation in the development of antibodies and reagents for biomedical research. She is a true leader in her field, and her generosity today speaks volumes about her commitment to the next generation of scientists. This professorship is the ultimate tribute to the power of mentorship. Nan has shared that she had multiple mentors at WashU, and today we honor her relationship with Dr. Frieden. Carl has been an extraordinary mentor to so many, and Nan's decision to establish this professorship in his name underscores the deep and lasting impact that faculty can have on their students' lives. When our alumni go on to great things, as Nan has done, and choose to give back in a way that uplifts their mentors, it is one of the greatest testaments to the WashU experience. This professorship will not only celebrate Carl's legacy, but also support future generations of scientists, ensuring that the kind of work that Carl dedicated his life to continues to thrive. You will soon hear more about Carl's remarkable career and impact from Dr. Ben Garcia. His words will help us appreciate just how significant Carl's contributions have been. From my personal experience, when I first came to start my own laboratory here at WashU in 1986, Carl was then the interim chair of the Department of Biochemistry. For me, at that time, he was one of the giants leading this school that was dedicated to advancing basic science and better understanding of human disease. My perception, now living indelibly in the recesses of my memory, was that he was one of the executive faculty striding in the clouds above. And that was amplified while I was here when it was announced that he was elected to the National Academy of Sciences, something that is only possible for the highest in achievements in science. So much of what we need to understand about human disease comes from work on the basic understanding of the allosteric behavior of enzymes and the folding and assembly of proteins during biogenesis, and how that is disordered in human diseases like Alzheimer's disease. This has been the focus of Carl Frieden for years on our faculty, and it has been the epitome of the world-leading impact of WashU science, how going deeper into basic science leads to new and better ways to treat human disease. Carl's close friend, Roy Vagelos, could not be here with us tonight, but he wanted to share a few words about Carl on this momentous occasion. And if you will, I'd like to read his letter. We just had Dr. Vagelos here on Saturday to celebrate the MSTP, and it was so marvelous to see him and hear him and to think of the impact that he's had as someone who came from WashU. Dear Carl, congratulations on the honor of the creation of the Carl Frieden Distinguished Professorship at Washington U. Your name will henceforth be connected with a distinguished professor of biochemistry and molecular biophysics. I know no one who has done more for Washington U in exploring important scientific questions than you. You have studied enzyme kinetics and mechanisms and protein folding. You developed the concept of histolytic enzymes. Did I pronounce that right? More recently, that's an old word, Carl. More recently, you have focused on ApoE4, working to understand the structure and function of this important protein that is involved in the neurodegenerative diseases, particularly Alzheimer's disease. With better understanding of this protein, drug discovery may be facilitated to control this dreadful disease. You were the first person, this is Dr. Vagelos, you were the first person that I met when I first visited the biochemistry department. I was immediately impressed by your great intelligence. After I joined the department, we, along with Louis Glazer, had lunch together every day. I was struck with your encyclopedic knowledge. I watched as you ate a peanut butter and jelly sandwich every day. I wondered whether that was the source of your fantastic intelligence. Being a scientist, I wanted to test this hypothesis, parentheses, a clinical trial. So I ate PB and J, that's actually how Roy wrote it, PB and J sandwich every day for a month. Unfortunately, I stayed pretty much the same. I did not get any smarter, and if you have any better ideas, let me know. You have been a great researcher and teacher, but you also agreed to chair the department on several great occasions. You were a great chairman, no surprise. Most of all, you have always been a good friend. Keep up whatever makes you what you are. Best wishes, Roy Vagelos. Nan, on behalf of Washington University, thank you for your generosity and vision that has made this professorship possible. Your success and your dedication to give back are an inspiration for us. And Carl, we celebrate you today as a mentor, scientist, and friend who will continue to shape this institution. And to all of you, we thank you for sharing the moment. And now I would like to ask Chancellor Martin to join me in bestowing the medallions on Nan and Jean.
You're supposed to clap. So Nan, Jean, we are so happy that you were able to spend some time with us. This gift is an extraordinary investment in the legacy of science at this university and all that it means for human health. So I have a shadow box for each of you, and so I'll present them one at a time. Nan, the first one is for you. The medallion reads, the Carl Frieden Distinguished Professor, in grateful appreciation, presented to Nan Jiang, March 12th, 2025. Nan. Jean, I have one for you too. We're really grateful for the time that you've spent here with us, getting to know us better. The medallion also reads, the Carl Frieden Distinguished Professor, in grateful appreciation, presented to Jean Lay, March 12th, 2025. Jean, thank you for your investment. Let's go over here.
So Nan, I think it's time for you to give some remarks.
Esteemed Chancellor Martin, esteemed Dean Perlmutter, esteemed Department Chairman Garcia, distinguished guests, faculty members, and staff and students, dear Dr. Frieden, dear Mrs. Frieden, and family, ladies and gentlemen, good afternoon. Today we gather to celebrate this special moment of the history of Washington University, the installment of Carl Frieden Distinguished Professorship, and also to honor Dr. Frieden, a remarkable individual whose contribution to science, education, and mentorship have left an indelible mark on all of us. Dr. Frieden has dedicated over 70 years to advancing our understanding of the molecular world and nurturing the young scientists. In 2029, I came back to WashU campus after left here for 30 years, and I visited Dr. Frieden. He met me in his lab. He was writing a paper, and he was 90 years old. And 5 years later, in 2024, I came back to visit Dr. Frieden again. He met me in his lab. He was writing a paper. And just a few months ago, I received an email from Dr. Frieden in which he wrote, "Hi Nan, just happened to be looking at the computer. My current project is to delineate the differences between ApoE3 and ApoE4, and I think I'm getting close to understanding why they differ." It is amazing, it is also rare to find someone who, after so many years, remains so deeply engaged in their work and continues to contribute to understanding the science. I had the immense privilege of working under Dr. Frieden as a postdoc researcher from 1991 to 1993. During this time, I was profoundly influenced by the kindness and the patience. Dr. Frieden's approach to mentorship was one of the gentle kind. He never raised his voice. I have never heard him raising his voice. Yet his authority and the respect he commanded were evident to all. His ability to foster a collaborative and a supportive environment in the lab is something I strive to emulate in my own career. Beyond his scientific achievements, Dr. Frieden loves art and has museum-quality collections. We had some lab meetings in his house. I was struck by the breadth and the deep collection he has. And this reflects his appreciation for beauty and creativity. This passion for Art is yet another facet of his multifaceted interests, showcasing his ability to find joy and inspiration in various aspects of life. Dr. Frieden, your tireless dedication to science, your mentorship, and your kindness have touched the lives of countless individuals, including myself. Your legacy is not only in the scientific research you have conducted, but also in the many scientists you have mentored and inspired. We are all better for having known you and worked with you. Thank you, Dr. Frieden, for your extraordinary contribution and for being a beacon of excellence and humanity in the scientific community. We celebrate you and today, and we will always celebrate you through the creation of the Carl Frieden Distinguished Professorship. I'd like to acknowledge Legion Foundation, through which the gift was given. The president of the foundation, Dr. Jin Lei, graciously accepted the invitation to come to St. Louis with me. I'd like him to see where I studied, where I did the research, the buildings, my mentors. And I also wanted to thank Jean for being a role model of giving back to the community. Lastly, I'd like to express my heartfelt gratitude to WashU Advancement, Will Arvin, and the team for facilitating and organizing this wonderful event so everyone shares the joy of the establishment of this professorship. Earlier today, we met several faculty members and visited the labs. I was very impressed. All I can say is that I'm so proud of WashU, so proud of the impactful programs, going on the campus, and so proud of the WashU people. And also, I'm so proud of being a WashU alumni. We share the same passion of goal, which is to translate research to therapy for better human health and human life. Thank you very much.
It is now my pleasure to introduce Dr. Ben Garcia, the Raymond H. Whittekoff Distinguished Professor and Head of the Department of Biochemistry and Molecular Biophysics at WashU, and he's tasked with telling you about Dr. Frieden and how he behaves in the department. Ben.
Good afternoon, everyone. Thank you, Chancellor Martin, Dean Perlmutter, and especially to Dr. Nanjiang and Dr. Jean Lei, for being here with us on this really special day and for making it possible. When this day was being put together, I was told I have 15 minutes to discuss Carl Frieden's scientific impact over decades. Definitely a challenge, but it's an honor and privilege to be here today to give this scientific overview of a scientist whose work has had an immensely profound impact on our understanding of protein and enzymatic mechanisms important in various areas of biochemistry. Carl Frieden, over the course of his distinguished career, has contributed significantly to the study of protein kinetics, enzymology, protein folding, computation, and structural biology, laying the groundwork for advancements in our fundamental understanding of how biological processes operate at the molecular level. Fundamental work that has developed theories and experimental knowledge to answer the most challenging scientific and biomedical problems. Carl was born and raised in New Rochelle, New York. He received his BA in chemistry from Carleton College and his PhD in chemistry from the University of Wisconsin under the mentorship of Robert A. Alberti, which started him on a lifelong journey and research interest in enzyme kinetics. In 1955, Carl joined the Department of Biological Chemistry here at the Washington University School of Medicine as a postdoctoral fellow to work with Dr. Sidney Velek. There, he published 3 papers as the sole author, something that is just unheard of in the scientific world. And then, after only 2 years as a postdoc, he was immediately hired by Dr. Carl Corey as an instructor in 1957, promoted to assistant professor in 1960, and then promoted to full professor in 1967. Just truly remarkable ascend from instructor to full professor in such a short time. For those who may not be as familiar with his contributions, which I don't really think there's anybody here in this room that is, Carl's research has been instrumental in unraveling the complex mechanisms that govern protein dynamics, folding, and function. Proteins, as you know, are the building blocks of life. They carry out many different types of biochemical reactions and other types of processes, such as transporting oxygen in the blood. Their structure, but also importantly, just as importantly, their kinetics are important to dictate how they function. I think Carl said it best in a reflection he published in the Journal of Biological Chemistry when he said, "We can determine all the interactions that are present in the cell, but unless we know the time dependence of these interactions, we will never figure out how the cell works." That was the great challenge that Carl set out to conquer. Carl's work has provided invaluable insights into the way proteins carry out reactions, fold and associate, and how these misfolding disorders can lead to disease. His career has spanned several decades, during which he has been a dedicated researcher, mentor, insightful collaborator, and wonderful colleague. As a professor at Washington University in St. Louis, he has shaped the minds of future scientists while pushing the boundaries of biochemical and biophysical research. His dedication to education, training, and research exemplifies the role of the scientist as both an explorer of the unknown, but also the guide for the next generation. It's evident that his mentorship has touched many lives. In fact, that's why we're here today, due to the influence he's had on one of his former postdoctoral trainees, Dr. Nan Jiang. More specifically, Carl has made seminal contributions to our current understanding of protein and enzymatic processes. During the course of his scientific career, he has investigated the fields of enzyme kinetics, mechanisms, especially of allosteric enzymes, protein polymerization processes, mechanisms of protein folding, and the behavior of intrinsically disordered proteins, just to name a few. I'd like to highlight some of his career research contributions and breakthroughs now. As Carl started his lab at Washington University, he was interested in studying protein-protein and protein-substrate interactions, especially in an enzyme system. And a perfect candidate appeared to him, the bovine liver glutamate dehydrogenase enzyme, GDH. In a series of groundbreaking publications published in the Journal of Biological Chemistry, Carl explored how different coenzymes, including diphosphopiridine nucleotide, DPN, and triphosphopiridine nucleotide, TPN, and their reduced forms, influence the enzyme's association, dissociation, equilibrium, and catalytic function. Using ultracentrifugation, Carl demonstrated that these coenzymes significantly affect GDH's sedimentation coefficient, which correlated with its catalytic activity. Furthermore, these findings suggested that the enzyme association enhanced enzymatic function, while dissociation leads to inactivation. Notably, the reduced form of DNP at high concentrations induced enzyme dissociation, leading to reduced activity, whereas DNP in its unreduced form promoted enzyme association and activity. The results established for the first time a direct relationship between GDH's structural state and its catalytic efficiency, indicating that the enzyme must be in an associated form for optimal activity. His work provided fundamental insights into enzyme regulation through coenzyme interaction and highlighted the importance of enzyme association in the metabolic processes. With this and further studies, Carl established the idea that the association of a protein with itself as a multimer can alter its activity, something that everyone takes for granted now and did for many years. But his work showed that the concentration-dependent association of glutamate dehydrogenase influenced its interaction with the purine nucleotides GTP and GDP as inhibitors and ADP as an activator. His key findings indicated that at higher enzyme concentrations, GDH underwent association forming a polymer, which altered its nucleotide binding properties. In contrast, at low enzyme concentrations, no polymerization occurred. Carl presented a rigorous theoretical model for the enzyme kinetics when the total enzyme concentration was comparable to the dissociation contents of a ligand, and validated this theory with a chemically modified GDH, which retained normal activity but could not polymerize. This work demonstrated that protein-protein interactions play a broader role in enzymatic regulation, particularly in allosteric enzymes, where they self-associate and this affects ligand binding. Given that GDH exists at high concentrations in mitochondria, his work here has direct physiological significance in cellular metabolism. Carl went on to introduce the hysteretic enzyme concept. I'm glad you asked how to pronounce that because I wasn't sure. In a landmark publication in the Journal of Biological Chemistry, Carl postulated that these hysteretic enzymes, hysteresis is Greek for lagging behind, were enzymes that respond slowly to rapid changes in ligand concentration. Unlike traditional allosteric enzymes that quickly adjust to metabolic signals, these enzymes exhibit a lag in response to slow conformational changes, ligand displacement, or polymerization processes. Carl suggested that this delayed response played a crucial role in metabolic regulation, providing a buffering effect that stabilizes intermediate metabolic concentrations in complex pathways. Experimental data from various enzymes, such as phosphorylase A and his favorite GDH, illustrated these behaviors with response times ranging from seconds to minutes. His methodologically developed kinetic equations to describe these processes and demonstrated that time-dependent enzyme activity can be mathematically modeled across different conditions. Carl postulated that the most likely mechanism occurring is enzyme isomerization as a primary driver of hysteresis. This mechanism is characterized by equilibrium between different enzyme conformations where the transition between forms happens to be much slower than the substrate turnover. Carl argued that such enzymes could regulate flux through pathways in a time-dependent manner, ensuring stability in response to fluctuating metabolic levels, emphasizing its potential significance in metabolic control. Carl also centered efforts on understanding actin polymerization as well. He investigated the mechanisms of actin polymerization induced by magnesium ions and demonstrated that magnesium binding to monomeric G-actin induced a necessary conformational change before polymerization could occur. Carl also revealed that the process was influenced by the presence of calcium ions, which competed with magnesium for the same binding sites and needed to be displaced before conformational change could occur. His work identified two magnesium binding sites on G-actin, a high affinity site that triggers a structural change, and a low affinity site required for polymerization. Polymerization followed a nucleation elongation model where once a trimer forms, actin filaments elongate by adding monomers. Experimental data and computational simulations supported this model, and he concluded that magnesium binding was essential for actin polymerization with a two-step activation process. These findings refined the understanding of actin dynamics, which are crucial for cellular processes such as motility and cytoskeletal organization. To further many of these types of studies I've just described, Carl established the use of kinetic modeling for the analysis of the full length of a progress curve. Previously, one would simply take the slope at the initial time point, initial velocity, or at the inflection point of a progress curve. However, Carl knew that using all of the information in the progress curve would be much more powerful and insightful. He developed the kinetic simulation, or KinSim program, a huge technological advance. KinSim was designed to simulate full-time course kinetic reactions, overcoming limitations of traditional initial velocity methods. This allowed for better analysis of non-steady state reactions, even capturing features such as bursts and lags. This program was applied to various enzyme kinetics, chemical reactions, and protein-protein interaction problems. He then turned KinSim into FitSim, a computer program that automatically fit the set of parameters to full-time progress curves for a family of reactions performed over a range of concentrations. This approach was extremely powerful for testing and excluding classes of models, as well as providing kinetic rate constant values that were otherwise unobtainable. Carl also became interested in another major scientific problem, the mechanisms of protein folding. To investigate this, he pioneered technological advances to monitor folding and conformational changes of proteins. This involved incorporating fluorine-labeled amino acids into proteins and creating a method of doing stop flow fluorine 19 NMR. In one study on the rat intestinal fatty acid binding protein, Carl analyzed the structure changes during unfolding and identified a previously undetected folding intermediate with this new sensitive approach. This intermediate persisted at high denaturant conditions, suggesting that it is more similar to the unfolded form than the native structure, guiding beta sheet formation in the process. This results challenged previous optical spectroscopy findings, which failed to detect these folding intermediates and demonstrated the sensitivity and power of the approach. More importantly, the findings contributed to a broader understanding of protein folding pathways, particularly for beta sheet proteins, and suggested that initial hydrophobic interactions played a key role in guiding structural formation. This was complemented by key work performed by one of Carl's postdocs, Dr. Nan Jiang, who used site-directed mutagenesis into the intestinal fatty acid binding protein to analyze how these substitutions affected protein stability, folding, and reactivity towards sulfhydryl-modifying reagents. Nan and Carl found that some cysteine residues reacted faster in the native state than in the unfolded state, suggesting structural flexibility in these regions. Together, they showed that conformational changes near the fatty acid binding entrance establish rapid thiol reactivity, which could be crucial for protein-ligand interactions and folding mechanisms. To further investigate the protein folding problem, Carl also worked to determine the folding mechanism of GROEL. GROEL is a bacterial chaperonin that assists in protein folding by preventing aggregation and facilitating the correct folding pathway. GROEL contains a large cavity where substrate proteins bind and are properly folded. Carl determined that GROEL binds unfolded proteins by recognizing small unstructured regions, particularly hydrophobic segments. However, once inside the GROEL cavity, substrate proteins fold via the same kinetic pathway as in bulk solution. Early dissociation from GROEL may lead to aggregation, while late-stage dissociation results in the correctly folded protein. Contrary to previous assumptions, Carl argued that GROEL does not accelerate folding, but instead prevents aggregation and sequestering by sequestering folding intermediates. Studies on dihydrofolate reductase refolding showed that the kinetics of refolding are identical in the presence or absence of GROEL. Thus, Carl determined that the folding inside GROEL occurs through the same mechanism as in free solution, with GROEL primarily influencing whether a protein folds correctly by modulating the probability of aggregation. Carl's most recent research investigations have focused on the mechanism of aggregation of proteins related to neurodegenerative diseases. He's taken on experiments to understand the properties of apolipoproteins, such as apolipoprotein E's, the major risk factor for the development of late on-stage Alzheimer's disease. In particular, Carl has explored the structural differences between the ApoE3 and ApoA4 isoforms. These isoforms only differ by one single amino acid, an arginine at position 112 for ApoE4 versus a cysteine in ApoE3. Additionally, ApoE4 is a major risk factor for late on-stage Alzheimer's disease, whereas ApoE3 is neutral in this regard. Using site-directed mutagenesis and biophysical approaches, Carl identified structural variations between ApoE3 and A4 beyond the immediate site of the single mutation. He found that regions in both the N-terminal and C-terminal domains of the proteins are affected, influencing lipid binding and oligomer formation. Interestingly, the rate constants for the monomer, dimer, tetramer dissociation were similar for ApoE3 and ApoE4, indicating that the observed structural differences do not significantly impact this equilibrium. However, the specific regions involved in association overlap with those structurally affected by the ApoE3, ApoE4 differences, which could impact interactions with ligands such as amyloid beta. By understanding these differences, Carl's work in this area provides a base for designing targeted therapeutics aimed at reducing ApoE4-associated risks for Alzheimer's disease. Carl has been heavily honored throughout his career for many of his scientific accomplishments and leadership. Among several awards, he has been awarded the Peter Raven Lifetime Achievement Award from the St. Louis Science Center, an NIH Merit Award, the Christian B. Affinson Award from the Protein Society, He was elected to the prestigious National Academy of Sciences in 1988 and to the American Academy of Arts and Sciences in 2004, two of the top honors that a scientist in the United States can achieve. Lastly, he has been honored with the Carl and Gertie Corey Faculty Achievement Award and the Second Century Award from Washington University as well. Carl has served Washington University and the scientific community with much dedication. He served as director of our MSTP, Medical Science Training Program, from 1986 to 1991, helping make the MSTP program one of the best in the country. Carl has twice stepped up to serve as interim head of our department from 1986 to 1989 when Louis Glazer left, and again from 1997 to 2000 when Gary Eckers left, before permanently taking over as a Witkoff Distinguished Professor and Department Head from 2000 to 2005. He not only maintained the quality of the department, but enhanced it. Carl has served on the editorial boards of several journals, such as the Journal of Biological Chemistry, Biochemistry, Protein Science, and the Archives of Biochemistry and Biophysical Journals. He has also served on multiple NIH study sections and several executive committee positions within the American Chemical Society, the American Society for Biochemistry and Molecular Biology, and the American Society of Biological Chemists. Carl has also contributed to our community as a whole through music and art programs as well. In 1975, Carl initiated and ran a program called Music at Moore, in Moore. That invited musicians from the St. Louis area, including many from the St. Louis Symphony, to present a one-hour program in the Moore Auditorium. There were 3 to 4 concerts a year, and the program ran for a decade and invited all members of the medical school to attend and was quite successful. In 2000, Carl initiated and ran a program to exhibit artwork around the medical school campus as well, and there many exhibits were shown over a span of 5 years. I hope this brief overview has given everyone a small glimpse into the sustained and impactful career of Carl Frieden. His meticulous work in the areas of enzyme kinetics, protein folding, and pursuing their mechanisms have provided clarity on the forces that drive protein dynamics and structure and the consequences of how errors in this process are even linked to human disease, such as Alzheimer's disease. However, beyond his scientific achievements, Carl has been an ideal role model in the scientific community. His colleagues and lab members alike speak of his intellectual rigor, his curiosity, and his generosity in sharing knowledge. He embodies the very essence of what it means to be a scientist, not just seeking answers, but inspiring others to ask better questions. So today, as we gather to celebrate his accomplishments, his career, and his influence on a multitude of scientists and scientific fields, let us take this moment to appreciate the dedication, brilliance, and passion that Carl has brought to the biochemical field and to Washington University. His pioneering contributions will continue to shape the scientific landscape for years to come, and is with this great admiration that we honor him here today. Congratulations, Carl. You highly deserve this honor and recognition. Thank you.
So now, if I could ask the Chancellor to come forward, and Carl, and Ben too, and we want to award you the shadow box.
Professor Frieden, we are in awe, not only of your scientific achievements, but all that you have done at this university for generations and for all of the students and trainees that you have mentored. What a wonderful honor to have your name permanently bestowed on this distinguished professorship. So it's my honor to present this shadow box to you. It reads, the Carl Frieden Distinguished Professor. In grateful appreciation, presented to Carl Frieden, March 12th, 2025. Carl, congratulations.
Well, I hardly recognized myself with that, with what Ben is talking about. But I wanted to thank you all for coming and talk a little bit about what we've been doing and what we've done in the past. And I want to particularly say that I've been extraordinarily lucky in terms of being here at Washington University. I couldn't have had a better place to do the experiments that we did. And I want to tell you a little bit about the enzyme that Ben mentioned, and to show you how lucky you can be. So when I started and was hired, I had to figure out which protein I wanted to work with. And I decided that I would work with a protein called glutamic dehydrogenase. And my family knows this story. Because the reason I was interested in it, it was the only enzyme, and it still is the only enzyme that uses both coenzymes. In other words, NADP and NADPH. And I wanted to know why. And so the first experiments that we did were just determining the kinetics and so forth. And I decided that what I wanted to do was to find out whether the two sites for different coenzymes were the same or not. So the idea was to add an inhibitor which is common to both substrates. And we did it. And the reaction went faster. And so this is not supposed to happen. And this was well before the term allosteric became popular, which was maybe 5 or 6 years later. But this single observation led to an enormous amount of discussion about how enzymes work. And I should say that at that time, which was 1957, there wasn't really much known about enzymology. So I was lucky to have found this observation. And because of that, we tried to develop as Ben mentioned, a system which would discuss the time dependence of enzymatic reactions, because one of the things that happened was that there was a time dependence in some of our reactions. And I don't think you will recognize what the computers were at that time. The one that we used was called a VAX, and it was the size of about 3 refrigerators, large refrigerators, and it was pretty slow. And so Bruce Gottschalk and another developed a program which would measure the time dependence of conformational changes. That program was available to anybody. And what happened was that a friend of mine, Ken Johnson, took up that program and started to make things better in terms of time dependence. And he runs now a business, which you may have heard of, which is called Kintech. And that was based on the computer programs that we had developed early, but it's much faster. Now it happens, you can run a Kintech experiment just as fast as picking up your hand. And so that was very lucky. And that kind of luck has sort of followed me through all of these experiments and so forth that Ben has mentioned. For example, another example, we undertook an examination of very important enzyme phosphofructokinase. And what people had known, or was known, was that the activity was not proportional to the concentration of enzyme. Well, that's not supposed to happen. And the reason for that happening, as we found, is that you had to recycle both of the products, which were allosteric effectors, in order to get the correct dependence of the enzyme concentration with the reaction. Those experiments, which led to a lot of papers, really were critical to the development of what we've been doing. And that is to look for things which you think you can provide an answer and try and find a way to do it. Ben also mentioned the project that we're doing now, which is to look at the difference between ApoE3 and ApoE4. And I think we have come to conclusions about those, and we're using methodologies that people really hadn't used before in a sense of distance measurements as well as the other properties. But it's been, being at WashU has been a fantastic job for me. I have to appreciate everything that happens in this school, that you can talk to anybody about your work and they will be happy to talk to you. So I thank you all for coming, and I appreciate this award which is just incredible, I think. So thank you very much.
So I think we have Dr. George Rose who's going to make some comments. Dr. Rose.
So thank you for this opportunity to say a few words about a very cherished friend. I will make some comments about Carl, but we all know the same person. So in the previous talks, you've heard the same comments, maybe with slightly different words. But let me tell you the bottom line. So if you forget all of what I have to say, the one thing that I want you to remember is really the bottom line of what I have to say. So let me start with that. I flew in from Baltimore yesterday, and this morning I went to Carl's office to spend an hour with Carl. I hadn't seen him in a couple years, and we exchanged a few remarks that old guys tend to do about mortality, and then after about 10 minutes, Carl said, "Okay, so now I have to tell you what we're doing with apolipoprotein." And we moved our chairs closer to the computer, and Carl brought up images of the structure and began by talking about just what he ended with now. Well, we measured distances, and you could tell from his talk that Carl is a natural-born teacher, and that's what we talked about. And that's Carl. But I will say some formal remarks. That's what I've been asked to do, and it's an honor to do that. So Carl arrived here at WashU in 1955. It was an especially propitious time to be doing what he's doing and what he has done for the past 70 years. 4 years before, Linus Pauling had proposed the his model of the alpha helix, which was almost immediately confirmed in X-ray crystallography structures that were going on at the time at the MRC in Cambridge. 2 years later, and 2 years before his arrival, was the Watson-Crick structure of DNA. And the chairman of the department at the time was Carl Corey. Two Carls are better than one. It was a perfect time to be arriving. The structures of proteins were just in the offing at that time. It was a great time. It was the start of structural molecular biology. It was a great time to be doing that kind of work. It seems especially poignant today to think back at those times when there was such excitement and such open-endedness and such a sense of discovery. And we think of that today in today's climate. So what Carl wanted to know, you know this, because you've heard it again and again, and just from Carl, is he wanted to know how things work. How do proteins work? How do enzymes in particular work? And his great insight, which he brought with him from the beginning, and which has been the insight that's guided his work throughout, was the insight that you can't simply look at the structure of the protein and figure out how it works any more than you can take a picture of your car's engine and know how your car works. The structure alone doesn't do it. You have to do kinetics. And you can't really know how it works until you've done a proper kinetic experiment. And Carl has spent his professional lifetime in kinetics. In kinetics, not only in his wet chemistry experiments, but in modeling as well. And another aspect of what he's done is that at the time he arrived, computers, as he said, computers were pretty elementary in those days. Well, I had at the beginning a VAX 11780, and I thought it was the be-all and end-all of computers at the time. But in fact, my cell phone has lots more memory and works a lot faster than my VAX 11780. Nevertheless, computers were made for biology, and Carl was a pioneer in that as well. So Carl has began to use enzyme kinetics to dissect the mechanisms of enzymes with great success, and he was a pathfinder in it. And you know all that because you've heard this in capsule and you've heard it in detail, and it has been transformative in every respect. The other thing I should say is that at Washington University, Carl has not merely made enormous scientific contributions, but he's made contributions to the university as a whole. He's stepped up to the chairmanship a couple times when called. He has been a contributor to the culture of Washington University in arts and in music. And he has also been an exemplar. He's won the most prestigious honors and awards. You've heard some of their names. But the other thing about Carl is he has not been a slave to ambition. One of the things we admire most about him. I think the other thing that I would say is that I've always thought that Carl was immortal. I still think that. There's no evidence to the contrary. He never seemed to age. There was a picture of him, his portrait that hangs in the department. I went and looked at it recently to see if that was aging. No, it wasn't. I mean, it's conceivable he has a picture in his attic, but I don't know. It is so wonderful, this celebration that you have organized for him. I want to thank Dr. Zhang for what she's done. I know about the protein she worked on, IFABP, fascinating protein. It is great to be able to pick up on the work that people have done. There's a continuity in this field, and there's a great curiosity and intrinsic inquisitiveness about how things work. Aristotle said, all want to know, and nobody wanted to know more than Carl Frieden and the people who have been his students, and he has inspired that in so many people. So our thanks to you, Dr. Zhang, for what you've done to honor Carl. You've left this permanent mark. And I would close by just saying that Carl has made a truly meaningful difference in the lives of so very many people, including me, especially me. Thank you, Carl.
Thanks so much, Nan and Carl, for making this happen for us. And everybody, please join us for the reception outside.